Today I'm going to talk about quality control and describe how an extensive quality control program used during the production of medical devices for IVF can help you improving your embryo culture conditions. Quality can be discussed in many ways and on several different levels. Today I will focus on toxicity and why it's particularly important to use safe and non-toxic products during gamete handling and embryo culture. As you most likely are aware of, the human embryo is extremely sensitive to stress and surrounding factors. In fact, the embryonic stage is the most sensitive stage of the whole life cycle. It is therefore not difficult to understand that quality and quality testing is of specific importance during handling and culture of embryos. I will divide my presentation into 4 sections or areas. I will start by talking about stress and toxicity and what stress can do to a human embryo and the clinical relevance of this. I will then describe the need for an extensive quality control program during manufacturing of medical devices for IVF. What shall a company do to provide safe, non-toxic products of high quality and high lot-to-lot consistency? I will specifically talk about the mass embryo assay and describe why there is an obvious need for a sensitive and reliable toxicity test. Finally, I want to talk about labware and use Vitrolife Labware as an example of the value of using an accurate quality control program during the manufacturing of medical devices. My intention today is to show you how quality testing can result in quality that makes a difference for you. So let us start with some basic facts regarding the safety of gametes and embryos during in vitro culture and handling. We know that during IVF we are exposing embryos to stress. In vitro culture, by definition, is stress. And there are a lot of different potential stress factors during in vitro culture. Stress is cumulative, which means that the embryo is exposed to the sum of all stress factors during the whole culture period. Stress reduces viability and results in lower clinical outcome. The more an embryo has to adapt to its environment, the more its viability is compromised. Obviously, the ability to minimize intracellular stress is a significant factor in being able to maintain embryo viability in culture, and is also a significant factor in creating high pregnancy rates. What does stress do to a gamete and an embryo? What do we actually see? Well, stress can result in reduced fertilization rates, impaired embryo development, seen as reduced cleavage rate, reduced development of blastocyst, or altered blastocyst differentiation. It can result in altered metabolism or altered gene expression. Altogether, this results in a reduced viability, reduced implantation rates, and in the worst case, it can lead to birth defects. This slide shows some of the factors that we know can affect embryo viability and subsequently the clinical outcome during IVF. In order to be successful during an IVF treatment, you have to secure the quality of the entire IVF procedure, which means that you need to control all these variables and all other variables present in your lab or in your clinic. I personally like to visualize an IVF treatment with a chain, where each of the factors listed in this slide form a piece of that chain. I guess you're familiar with the expression, no chain is stronger than the weakest link. Even if everything in your lab is optimized and under control, one suboptimal factor can be enough to lower your results or even ruin it. One toxic culture dish, for example, can be enough to destroy the whole chain. So, one question that people working within the field of IVF should ask themselves is there a need for testing medical devices prior to clinical use in IVF laboratories? The answer is, without any hesitation according to me, the answer is yes. If you answer no to this question, I think you belong to this type. I dare to show this slide since I have performed thousands of mass embryo assay as a toxicity test over the last 20 years. In our laboratory, we have routinely performed this assay on both raw materials and final products used in IVF, including disposables, contact material, chemicals, media, etc. There is a surprisingly big variation in quality between different types of products, between brands, between lot numbers, and sometimes even within batches of the same lot. So how often do material or products fail in our toxicity test, in this case in our mouse embryo assay test? This slide shows data from Professor Gardner's laboratory as well as Vitrolife's mouse embryo assay lab. Both these laboratories test all medical devices and raw material before it enters the lab or are used in production. This is data collected over several years. As you can see, 1 in 4 of contact supplies, such as dishes, fails the test with a sensitive one-cell mouse embryo assay. That is 25%. This type of contact supplies is average labware used in IVF clinics. For oil, raw material, it is worse. There, 4 in 5 fails the test, which is 80%. Chemicals show similar failing rate as contact supplies, which is 25%. When you see data like this, it is important to highlight that a fail rate of 25%, for example, does not mean that the tested material is deadly toxic. The embryos are not immediately dying here. The embryos are affected by some suboptimal factor. So we're talking about suboptimal conditions. And since they are exposed to this suboptimal factor, they do not develop at the same level as a safe control and therefore should not be used. Well, these observations or these levels of toxicity are not only seen by Professor Gardner and by Vitrolife. In a study by Nice et al. From 2009, very similar results were reported. In this study, which was a 4-year survey, they evaluate medical devices used during IVF, and I found a concerning level of toxicity, referred to as reprotoxicity in this study. Let me show you some of the data. Totally 350 human sperm survival tests were performed. They tested 36 prototypes and totally 72 different brands of products. They divided products into 2 groups, products used for patient-related procedures and products used for gamete and embryo-related procedures. Out of the 36 types of contact material that were tested, 40% were toxic. 42% of the products used for patient-related procedures were toxic, and 10% of the products used for gamete and embryo-related procedures were toxic. So, to summarize this first section, there seems to be a need for more extensive quality testing of medical devices prior to clinical use in IVF. The question is, which medical device should be tested? Well, our experience is that all materials that come in contact with gametes and embryos, primarily or secondarily, should be tested before clinical use. Okay, I've now talked about how sensitive embryos are to various stress factors, including toxicity. I've also shown that commercial products available for IVF varies significantly in quality. The question is, what does it take to avoid toxicity and suboptimal culture conditions? What can a company that produces medical devices for the field of IVF do to guarantee the delivery of safe, non-toxic products? What shall be included in an extensive quality control program? I had listed on this slide 3 overall criteria that I consider a must for an extensive quality control program for medical devices used in IVF. First, quality and quality testing needs to be performed on several different levels during manufacturing, from raw material all the way to the final product. Secondly, a series of relevant test methods is required to detect all the various parameters that need to be controlled. And finally, there is an obvious need for a reliable, functional test that also works as a biological toxicity test. At Vitrolife, we perform quality control on mainly 3 different levels during manufacturing. Level 1, raw material testing. Level 2, testing during manufacturing, also referred as in-process testing. And level 3, final product testing. We place particular emphasis on raw material testing, such as medium components and contact materials. Testing of raw materials gives us a unique possibility to actually create high quality, since we can test a lot of different raw materials and then select the one with the best quality and use that in our production. I want to highlight that at Vitrolife, each single lot of raw material is tested and evaluated by stringent quality control test procedure. And it's important for you to know that the raw material is not only the chemicals or materials used in our products, it is everything used in production that gets in contact with the final product. So it's vessels from mixing media, the tubings, filters, bottles, caps, etc. Level 2. Testing during manufacturing or in-process testing is also very important for the quality of the end product, and so is the actual manufacturing environment itself. The final product testing is of particular value to you, I can guess, since this is the tests of the products that you are going to use in your clinic. The results from these tests, at least some of them, are what you see on the certificate of analysis for each product. As I said before, in order to obtain an adequate quality of medical devices used in IVF, it is essential to use a series of relevant methods for quality control. One test method is never enough to detect the various hazards that needs to be controlled. This slide shows some of the many tests we're using. I will not go into any details about these tests today. I will, however, focus on one of our tests, the mouse embryo assay, or MEA, as it's described and highlighted in this slide. Before talking about the mouse embryo assay, I want to summarize why we perform all this quality control and quality testing. Why do we spend all these resources on continuously testing our raw materials, our products, and our manufacturing? There are a lot of reasons, and I have listed some of them. One reason is to guarantee the function and efficacy of the product. If we develop a product for separating sperms, we have to test and make sure it's working for that. Another reason, and maybe the most important reason, is to increase safety, to minimize the risk for the mother, for the embryo, and ultimately for the baby. Achieve high lot-to-lot consistency, which will give greater possibility in consistent results in your clinic. Increase efficiency at the IVF clinic by providing pre-tested products or ready-to-use products. Save both time and efforts for you by using pre-testing products. Of course, it is to comply with regulatory requirements, and finally and hopefully to increase clinical success rate. Vitrolife mouse embryo assay. A prerequisite for an effective quality control program when manufacturing products used in IVF is the employment of a reliable and sensitive toxicity test. I will therefore now switch over and focus a bit on the mouse embryo assay. What is a mouse embryo assay, and what shall an accurate and sensitive assay look like? The mouse embryo assay is, as I mentioned before, a toxicological and functional assay. The safety and efficacy of culture media, media components, disposables, and other devices are determined by observing the in vitro development of mouse embryos exposed to the item to be tested. MEA is one of our most, or according to me, the most valuable tool for the identification of various toxic and suboptimal conditions. Although mouse embryo assay have been widely used for quality control in the field of IVF, with several reports on their beneficial effects, the mouse embryo assay has also been criticized for not being suitable and sensitive enough to detect suboptimal culture conditions for human embryos. There have been a lot of conflicting data regarding the use of the mouse embryo assay. However, it should be kept in mind that the outcome of a mouse embryo assay depends very much on the way the assay is performed, and I will come back to that. My opinion is that One could get almost any results with the Mouse-Embryo assay depending on how the assay is performed. And this is what I'd like to discuss now. The conflicting data regarding the capacity or limitation of the Mouse-Embryo assay can be explained by, well, several things. I listed some of them here. Type of media that has been used, supplementation with serum or albumin, stage of development used at the start of the culture, strain of mice used. The MEA is very complex. It's a complex assay with hundreds of variables, and the outcome of the assay depends very much on the way it's performed. Despite the complexity and all these variables, the MEA has never been standardized or harmonized, which is extremely important and strange. 1998, FDA decided that the Mouse-Embryo assay should be one of the special controls needed for medical devices used in IVF. So FDA made MEA more or less mandatory for IVF devices. But they never defined how it was going to be performed. Knowing that the sensitivity and accuracy of a Mouse-Embryo assay depends on several factors and that an assay can be performed in many different ways, it is very important for end users like you to understand the capacities and the limitations of specific Mouse-Embryo assays. Since there is no standardized method used by manufacturing companies, you must be critical to their MEA. Different manufacturing companies are using different types of Mouse-Embryo assays, and as a consequence they will use assays with different sensitivities. In order for you to be able to evaluate if a specific product is safe to use and has the quality needed for the embryo culture, it is important that you understand the how sensitive an assay is for that specific product. Furthermore, it's important to remember that there might be different reasons for why Mouse-Embryo assays are performed. In order to be able to sell medical devices in, for example, US, you have to perform a Mouse-Embryo assay on that product, otherwise you have to label it as non-embryo tested. What I'm trying to say is that there might be other reasons for performing Mouse-Embryo assays than testing for safety and toxicity. It can be exclusively for commercial and regulatory reasons. So back to the sensitivity. As I said before, the outcome of a Mouse-Embryo assay depends very much on the way the assay is performed. In this slide, I have listed some of the factors that affect the sensitivity of a Mouse-Embryo assay. All these factors influence the accuracy and the sensitivity of the Mouse-Embryo assay, and hence the outcome of the quality control test. Let me give you some examples of these factors. The mouse strain used as the source of embryos will affect the sensitivity and thereby the results of a Mouse-Embryo assay. The developing potential of embryos, including both the rate of development and sensitivity, have been reported to vary between strains of mice. Inbred strains and their F1 hybrids are less sensitive compared to outbred strains, which are more sensitive to environmental factors and actually quite difficult to culture. You get a considerably more sensitive assay when using embryos from outbred CF1 compared to F1 hybrid, as you see in this slide. In the graph to the left, you see culture, a control, and a paraffin oil that was tested using a inbred strain or an F1 hybrid. And there seems not to be a big difference between the control and the paraffin oil tested. If you do exactly the same test, but you use an CF1 instead, you see a bigger difference between the two products, and there you will be hesitant to actually use the paraffin in test. Most companies use F1 hybrid mice as embryo source in their MEA. For testing key substances and key raw material, it is recommended to use outbred CF1 strains for maximum sensitivity. At Vitrolife, we use the more sensitive strain for key compounds like amino acids, proteins, hyaluronans, and oil for the production of uv oil. Stage development used at the start of the culture is also important for the sensitivity of the Mouse-Embryo assay. It has been reported in numerous studies, and it is also our own experience that mouse embryos collected at the one-cell stage are more sensitive to suboptimal culture conditions than embryos collected at the two-cell stage. This is correlated with the critical steps of development that occurs at this stage of development. Another very important factor affecting the sensitivity of a mouse embryo assay is the medium used when testing raw materials, chemicals, and contact supplies. In general, the medium used shall be protein-free. The rationale for using protein-free medium is that serum or serum albumin can chelate potential embryotoxins, such as heavy metal ions, present in the medium. The presence of proteins would therefore hide any potential detrimental effects of the medium. Testing media should not contain other chelating agents as well, such as EDTA and amino acids. At Vitrolife, we have developed a specially designed test medium, a stripped down version to maximize sensitivity for medium components, contact materials, and critical devices used for media manufacturing, including our labwares. In this study, we have taken away all good stuff. No HSA, no amino acids, no EDTA, no vitamins, and a lot of other things. It's like pushing the embryo towards the edge of a cliff and then start the culture. If something is suboptimal, we will get an effect and we can measure it. Maybe the most important factor for the sensitivity and the accuracy of a mouse embryo assay is the endpoints used. Most mouse embryo assay protocols employ a single endpoint for assessment. Commonly, day 5 blastocyst development should be more than 70 or 80%. Vitrolife use multiple endpoints of development, and including several different and specific timings of scoring, blastocyst development, blastocyst cell number, and blastocyst differentiation. Let me give you a few examples here as well, and start with the timing of scoring. The rates of development shall be determined by scoring embryos at specific times during culture. As just mentioned, most embryo assays protocols employ single endpoint assessment. We score embryos at several time points. We score them at 9 o'clock day 3, 9 o'clock and 4 o'clock day 4, 9 o'clock day 5, and we score them at exact time points to follow development. It is evident that mammalian embryos can be cultured to the blastocyst stage in a variety of conditions. However, the resultant viability of such blastocysts can vary enormously. Blastocyst formation or blastocyst development is therefore a poor indicator of embryo quality and does not accurately reflect developmental potential. A more sensitive and quantity parameter is blastocyst cell number. Therefore, we do cell number counting on all our products and raw materials. And in fact, I think we are the only company using the multiple endpoints assessment, including blastocyst cell number. As I said before, blastocyst development or blastocyst formation is not the same as a viable blastocyst with high implantation potential. I usually use this slide to explain why cell number counting is an important part of a mouse embryo assay. This is data from an old study by Professor Gardner that shows that fetal development is proportional to blastocyst cell number, but not to blastocyst formation or not to hatching. This slide shows an example of what I'm talking about. At this slide, you see 3 mouse embryos or 3 mouse blastocysts. The 2 to the left have expanded and developed further than the one to the right. The one to the right look worse. If we were using blastocyst formation as our only criteria for evaluating embryo quality, the 2 embryos to the left are equal in quality and the one to the right is of poor quality. However, when we look at the cell number of these blastocysts, we see that the 2 embryos to the right are poor compared to the single one to the left. The embryo to the left has significantly more cells, which is a good indication for viability. Suboptimal culture conditions and stress slow down embryo development and the cell proliferation, and this can be measured as a lower total cell number at the blastocyst stage. So, what are the standards when it comes to mouse embryo assay? Well, regulatory authorities say that you have to perform a mouse embryo assay, full stop. But they don't say anything about how you should perform that assay. The industry standard states that the passing criteria is only to have more than 80% expanded blastocyst at 96 hours, which means to use an assay with a single endpoint of assessment. The Vitrolife standard exceeds both of these. As you can see, the passing criteria for the Vitrolife MEA is much more extensive. Vitrolife is using the multiple endpoints assessment, including blastocyst cell number. The product or material tested shall pass at every time point the defined specification for each stage. This creates a MEA that is more repeatable, controlled, and inherently more sensitive. If we then add the fact that we use specifically designed test medium to maximize sensitivity when testing contact material and raw materials, and that we use one-cell mouse embryo assay from inbred CF1 mouse strains for testing key raw materials, you can see that we are ahead of the industry, or the industry standard. Finally, I want to talk about labware and use Vitrolife labware as an example of the value of using an accurate quality control program during the manufacturing of medical devices. My question earlier was, can an extensive quality testing program result in quality products that makes a difference to you? As you remember from my previous slides, there are certain challenges with suboptimal labwares on the market. Vitrolife labware. Vitrolife has developed a series of labware that is specifically designed for IVF, produced for IVF, quality tested for IVF, and regulatory approved for the correct indication for use, for the processing of gametes and embryos that will be used in fertility treatment. These labware are extensively quality tested, which means they follow the extensive quality control program I have described today during this presentation. That includes raw material testing, in-process testing, and final product testing. It also means that our accurate and sensitive mouse embryo assay, as well as human sperm survival assay, have been used. The product range of labware covers the need from aspiration to transfer, which means that the products with the same high quality will be available for all procedures in the lab. No weak pieces in this chain. This labware also meets the regulatory requirement. I will come back to that later. Let us dig a little deeper into the extensive testing. I have described in detail our quality control program, our mouse embryo assay. The sampling procedure for each produced lot is of course important for the quality. Since each lot produced can contain thousands of products, there is a risk you will get a variation within the lot, a variation in quality. To avoid this, samples for QC testing is always taken from the beginning, from the middle, and from the end of production. When it comes to the exposure and extraction procedure during testing, the general principle is that the same exposure should mimic, the sample exposure should mimic the way a product is used at the clinic, according to the instruction for use. We follow the principle of worst case scenario to make sure that the products we test are properly exposed. The timing has to be long enough to guarantee that you extract potential toxicity if that is present. As mentioned earlier, we always use a specially designed test medium for these type of tests. That is to maximize sensitivity. Just a few words about the production of labware, since that has an impact on the quality, of course. The way you produce labware will affect the final product quality. In this slide, I have listed the most important factors that defines the quality of labware. These are the raw material, or the raw material quality that is used, the production process, the plastics and materials in the sleeves, and the sterilization method and the intensity used if we're talking about radiation. The temperature you use during production will affect the quality. A higher temperature will increase the production speed, but will also lower the quality. A lower production temperature, on the other hand, will lower the heat-induced stress. Some companies even use mold release chemicals to speed up the process, but this will definitely lower the quality of final products and should not be used. Finally, the sterilization process is very critical for the quality of the labware. This is most likely the single most important factor for defining quality. Too hard radiation will for sure create toxic labware. Sterilization should occur at as low doses as possible with a preserved sterility assurance level. I promised to get back to you about the regulatory situation. We say that Vitrolife labware meet the regulatory requirements. What do we mean with that? This is a heavy slide, but I will guide you through it. If we look at the title, it says clearly that IVD devices is exclusively intended for in vitro diagnostic and not for IVF. The majority of medical devices used in IVF procedures are classified as medical devices or general medical devices, and they are subgrouped into class 1, 2, 3. Not labware though. Labware is very often incorrectly classified as an IVD by the producers. In fact, most labware on the market are registered as in vitro diagnostic. That is okay, I guess, if that is what they're going to be used for. But if they're used for IVF, they are not used in the right way. The fact that labwares are incorrectly classified as IVD has certain implications. For most IVD devices, no independent third-party notified body needs to evaluate and approve the product or the manufacturing process. This means that labware classified as IVD is under far less control. If we read the standard from the MedDev, it says that the IVD is used in vitro for examination of specimen derived from the human body, and where such specimen is never reintroduced into the body. IVD devices are not intended to be used for the processing of gametes and embryos that will be used in a fertility treatment. So I summarized this in a somewhat lighter slide. What is then the difference between IVF certified and IVD certified labware? If we start with the IVD certified labware, unfortunately, most labware on the market used for IVD is classified as IVD. It is exclusively for in vitro diagnostic and not for IVF, which means you can culture gametes and embryos in this, that is not a problem, but you can never transfer them back into the body. If we look on the IVF certified labware, like Vitrolife labware, it conforms to Medical Device Directive for indication within IVF. Indications for use is for processing of gametes or embryos that will be used in fertility treatment. This means it will allow embryos to be reintroduced into the body to be used in embryo transfer. The key difference is in the instruction for use. I have now talked about toxicity in IVF, what shall be included in an extensive quality control program, and how this has been implemented for a series of labware produced by Vitrolife. What will happen if these are used in clinical IVF? Will we see a difference in the clinical outcome? In order to evaluate the efficacy of Vitrolife labware, we collected clinical data from 3 independent IVF clinics, one in Pacific region, one from Germany, and one from Scandinavia. The clinics used the labware according to their own standard procedure. This means that we collected real-life clinical data. This is a retrospective evaluation with historical control. So we have to, this is not a full study, that's why we call it evaluation. Interesting is that 2 of the clinics used embryo-tested labware as the control, and 1 of the clinics used a rinsed labware as the control. These are the results from Clinic A. That's a clinic from Pacific. This clinic does more than 90% single embryo transfer, and they reported data from both fresh and cryopreserved embryos transferred early or at the blastocyst stage. Most of them were at the blastocyst stage. As you can see, the results were consistently higher with Vitrolife Labware compared to the conventional Labware. If we look at the fresh all ages, which is numerically the biggest one, and that's what I want to focus on, we see that Vitrolife Labware, the clinical pregnancy rate was approximately 5% higher, indicating a relative improvement of 14%. There is also support for the improvement in the subgroups. However, the number of patients are smaller, and therefore I would be careful to draw any conclusions. This clinic performed mainly blastocyst transfer, as I said, and the clinic has used mouse embryo assay tested CE marked Labware as their control, but not mouse embryo assay tested by Vitrolife. In this case, they only tested the dishes. These are the data reported from Clinic B. This clinic does more than 90% single embryo transfer at day 2, day 3, a typical Scandinavian clinic. And they reported only their fresh cycles. Here we see approximately 4% units higher than conventional dishes, indicating a relative improvement of 12%. Control here as well was MEA tested certified dishes. Only dishes were tested. And finally, these are the results from Clinic C. Again, we see the results were higher with Vitrolife Labware compared to conventional Labware. Here we see approximately 7% increase compared to the conventional dishes. This slide is a summary of all 3 clinics. 3 independent clinics in different parts of the world with different protocols and regulations. They all get improved results by eliminating toxicity. As you can see, the results were consistently higher with Vitrolife Labware compared to conventional Labware in all clinics. With Vitrolife Labware, the clinic pregnancy rate was approximately 5% units higher in all 3 centers. I'm fully aware this is not a study, this is a retrospective comparison with historical control, but the fact that 3 clinics give similar results is quite interesting. So this is my last slide, and I want just to summarize what we talked about. I hope you're still there. I appreciate that you've listened. The conclusion from my presentation is that the use of an extensive and systematic QC profile for all raw materials, manufacturing process, and the final products can help end users to improve their culture conditions. Extensive toxicity testing should not be limited to the culture media, but should include all supplies that come in contact, direct or indirect, with gametes and embryos during the IVF procedure. Each single lot of raw materials and final product shall be tested and evaluated by stringent quality control test procedures. Particular emphasis should be placed on the selection and quality testing of raw material and manufacturing environment. And using an unbroken chain of quality products can help you improve your results. Thank you very much.